I would like to start by welcoming everybody to the 24th edition of the Jamalpur Alumni Association's virtual lounge session. We have, after a gap of about 5 months, we have a Simi who's with us, and we are very grateful to Professor Rashmi Chowdhary, wife of Nitin Chowdhary, 83 batch, who has very kindly agreed to talk to us on her journey from a homemaker to a scientist. It's a journey into research and beyond. Very impressive credentials. I'm sure all of you must have seen the notice. I think it's into microbiology and molecular and nanotechnology and all the latest terms that one can think of. We would love to hear from her. And with that, I would like to hand over the platform to Professor Rashmi Chowdhary to let us know of her journey from homemaker to scientist. Over to you, Professor Rashmi.
Thank you very much, sir, for my kind introduction. Uh, Thank you very much for your patience, everyone. Thank you so much, and namaskar. First of all, I would like to thank everyone for giving me this opportunity to present some of my experience. And although I don't think that I have achieved something very extraordinary, and it was just my, but all of you are like just like my family, so it is good for me to share whatever I have done during those few years. So thanks again. I'll take a very brief about what actually scientists do in the healthcare system. Actually, we are the backstage artists who are doing work, doing some manipulations, designing some research question, working on some hypothesis and developing certain strategies and testing. And when once they are approved, we give it to the frontline workers to test them or to implement them. So, but this particular form of backstage working is highly high rate of failures are there. There are more than 99% of failure, hard work, and doesn't give you strength, but it always require a lot of dedications to come up again next day to start right from the beginning, from the scratch. So with that, I'll begin my journey. So I was like leading a comfortable life, got married with a railway officer, and was started happily and moving around with him with from places to places. And at that time, I thought that railway is working with only four people who are very important, like station superintendent, guard, ticket checker, and driver. But when I got married, I come to know that, oh my God, it's a huge, huge organization. There are so many people, so brilliant people like you all are there who are working day and night to make the railway working so fantastic. And I also started myself, making myself comfortable in that atmosphere. I started learning new terms like CNW, box and depot, food plate infections, and hooter. What does hooter one means, and what are those yard derailments and everything. So I was like quite into that system and started, but somehow I used to miss my higher education, which was left somewhere because of this change of my place from one place to my father's place to my husband's place. But in meanwhile, I was like working with railway or women organization, and apart from that, I was also involved in many community based activities on my own. And for the railway also, I work a lot for the schools and their adult education and all. Then it was almost 10 years. Then we moved to a place which was full of institutes of national importance. And I somehow went to all these places, and I was like surprised to see that how to get through those national institute to study. But because of this 10-year gap, everything was changed. I was not in touch of what is going on that time on the science front. But still, I went to SGPGI, that is Sanjay Gandhi Postgraduate Institute of Medical Sciences in Lucknow. And while going around in many departments, I came up to a department of microbiology, and I met my future mentor there, Professor Dr. T. N. Dhol, and he was nice enough to meet me and say that, okay, if you are interested. Then I asked him a very simple question: how can I study here? And he just smiled and said that, okay, I mean, you are not in college, you are away from the studies for a long time. I don't know how you will set up here. But out of politeness and to see my enthusiasm, he said, okay, you can come and see our working in the virology lab for a few days. Because he thought that after 3, 4 days, she will run away. She has nothing to look forward. It was a highly containment laboratory. So he said, just sit at a place where we receive the samples with all attendants. I said, okay, that is also good enough for me. So I sat there, and that time I was so happy to be in that institute. And I was like, I was going there, and I was thinking that कि पता नहीं अब कल क्या होगा, हमारी ट्रांसफर हो जाएगी, आने मिलेगा नहीं मिलेगा. But I thought that, okay, whether I'm employed or not, I just like that atmosphere of that laboratory. It was so contagious that I could not say that I'm not going tomorrow. So I started going there, and then the days become month and months become year. And I gel with all the youngsters over there. They were very junior to me, but they also accepted me. But and I learned many techniques, and I started working there and learning. And basically, within a year, I was like expert in all the basic techniques which were being done there as a trainee. And this happened that one at the time in 2000, there was a WHO team which was visiting in our lab from Geneva, and they were like asking all the key questions to the staff. And I was standing somewhere behind the group of students and scientists, and they asked something, and they were all like, were not able to answer. And my mentor at that time, he thought that I know the thing, and he said, come here, and if you know, just why don't you explain it? I said, okay, sir. And then I explained them, and they said, okay, who is she? She's they that time my mentor said she's a trainee, and they said, oh, she looks good. She should be a mid manager here. And that day I was no longer an observer. I become a contributor in the lab. And then my mentor encouraged me to appear for an exam. I joined there as a senior research fellow. And that time I was like, that transition phase from a informal learner to formal scientist was taxing, because when I was coming to the lab, I didn't have any responsibilities. I was just coming and learning things. But then my journey started. I realized that it was such a testing for mind, body, and soul, that we were working on with the WHO at that time, and that period was dependent, the global polio eradication was dependent on UP and Bihar. I'll tell you a background about what was that. So, the polio was eradicated from European continent, Western continent, Australian continent. Only, of course, Africa and Afghanistan were like kept aside at that time because some war or anything was going on. But the Asian continent, where the polio was in a massive grade, was contributed by these two states of Uttar Pradesh and Bihar. So I was working there with the team of WHO, CDC, and NIH scientists, and we had lot of continuous discussions that how to cut down the time, because this particular thing is contagious, and it is like a oral fecal route contamination, water, or human to human transmission. So the time period for that time of identify a wild polio virus circulation was 21 days. But the high population density in UP and Bihar, 21 days is a good enough time to, if one kid get the infection, that kid can spread the infection to hundreds of other kids. So, the mopping up time, the oral polio virus implementation strategy, surveillance was not fitting in that 21 days time. So when we did lot of discussion on this particular problem of polio virus, I come up a idea that screening each and every person in high population density, going from door to door, it is just not possible. And since this particular enteroviruses where the polio virus come under that, it is a fecal oral route it follows. So can we test the sewage sample? If we test the sewage sample, we will actually screening hundreds and lakhs of people by checking the 1 liter of sewage sample. So that particular thing was very exciting for all the WHO and CDC scientists. Then also we discussed that can we shorten the time of testing or this from 21 days. So my team and in my initiative, we developed a PCR, that is integrated cell culture PCR. So that molecular testing has given us time to identify polio virus in 4 to 5 days, and that cut down the whole system. And we used to go from one place to other place once we get the signal that particular person or particular kid is from some interiors of Bihar or UP is detected, so that within no time the team used to go isolate that kid, and the all the areas of mopping up around used to take place. So that has played a very important role in polio eradication, and slowly from 2000 to 2008, polio virus, of course there are other things, but this particular thing. And when we tested this slide, you can see that when we did this, we could find out the wild polio virus transmission prior to its outbreak. So that there was one research setting where we test this thing. It was a UP, in which we found that the virus was detected in January in environmental sample in Lucknow. Although Lucknow didn't have any cases till particularly in that time, we were like flagged that thing that we got a wild polio virus in Lucknow and it has reached. And the team was all ready. Second, the first case we could find in Lucknow was 30th of June. And once we got that case in Bakshi Ka Talab on 30th of June, the team was prepared. They immediately isolated the kid, and all the mopping up rounds to his siblings and neighbors and everything was done. So there was only 9 cases which came up, and that outbreak was not never happened in that particular strategy. So for this study, I got a best paper award in a clinical virology symposium in Florida, and they appreciated this work, and it turned out to be the major work in the polio eradication, global polio eradication of the WHO. I got a travel award. We went there and I got an award for this best paper award. And basically, it was very much appreciated. Still, whenever there is an viral outbreak, my this paper and my this study is always quoted for lowering the testing time and also the how to control the outbreaks. Then because of my this work, I was very fortunate that I got the opportunity to work with the very eminent scientist of the Ivy League University. Professor Dr. Ian Lipkin, who was a director of Center for Infection and Immunity in Columbia University. He is known as a virus hunter, and he works with the pathogen discovery group with NIH. And they were working since many years, and they were always knew that there may sometimes bioweapons or some pandemic will definitely going to be there. So they were all prepared for this. Dr. Lipkin is known for virus hunter. That's a very funny name, but he has discovered more than 300 viruses since 2000 and onwards. That means those viruses were not new, which has emerged, but the technology he has developed is the next generation sequencing, and that has empowered him to find out the viruses which are hidden in water, in seawater, in sewage, in animals, or in zoo animals, or wherever there is a unknown etiology of the viral infection. They found it. So when I went there in Colombia, he was working on SARS-CoV-2, SARS-CoV virus, which was at that time was an outbreak in China. And then he was the person who was working on the second, after 10 years, there was the same group coronavirus outbreak as a MERS virus. And I was like, once I was talking to him, and I was just joking that I was like doing all those viruses with you, and the third virus, SARS-CoV-2, I work in India because of your training. So yeah, that time, and this postdoctoral research working in Ivy Lag was amazing experience, but it is on a very different tangent. I haven't worked anything like this in India ever. They work at the different platform. Their thinking, their planning, and everything is so different. But they are really hardcore people into the research, and they make you so strong while putting you on a different level of task. So those 3 years, I really worked hard there, and I learned so many things. Firstly, I work with the international platform where Chinese and other people, we are working together. I was working in the highly containment lab, that is a BSL-3 lab, and taking that training and working under those and doing all the high-end techniques was a lifetime experience for me. I have worked on many projects with them. We discover many new viruses. They are from the Banya viridae family. They are also mosquito-borne viruses in US. So, and published papers. And while working there as a research scientist, I got an interview call, and my conducted by UPSC, and I got selected. Of course, I applied for it, but that application was more or less for my Indian mentors. They forced me that why don't you come back and serve your country. So I said, okay, I'll apply for it. And when I got selected here, I joined AIMS Bhopal as assistant professor and the founding faculty of institute. So now the journey was started, and as a postdoc and the researchers, I started as a medical teaching and diagnostics, and taking up studies for MD students and PhD students. I was into the admin work, doing some accreditation work, but my research area was always on the front foot. I just love research. Now I have like I developed the BSL facilities in AIMS Bhopal. I am a key member of developing the BSL-3 facility in AIMS Bhopal, and doing and taking up many projects related to infectious disease from H1N1 to dengue to SARS-CoV-2. So my key area of interest is molecular virology, cancer biology. And with my present head of the department, I got so much of information and thing on the nanotechnology. So I'm now looking forward for targeted drug therapies, especially for very hard to do surgeries like brain tumors and all. I'm working on that. I'm also working towards a concept that whatever we do intervention in therapeutics, it should not be it should be minimally invasive. So I'm working towards that, and I have many projects related to cancer biology, especially cervical cancer and brain tumor. This study, I would like to take up as well, is very close to my heart. This was very initial project was done at for the vitamin D status. So I did this study was funded, and I did vitamin D to find out in adolescent girl. And I was surprised to see when I was doing this research, 90 more than 99% of these girls are hypovitaminosis D. I was taken aback as a shock of my life that these adolescent girls who are going to be a future mother are there are bones have no strength absolutely. And the second thing which we observe in these girls while doing the genetic profiling and other testing is that they are, because of the hypovitaminosis D, they are having secondary hyperparathyroidism. That is a side effect of hypovitaminosis. So what happened when vitamin D is very low, thyroid gland becomes so active, so it gets all the calcium from the bone. So these girls are uh, inhibit by going to the taking the sun rays, because that is the major source of vitamin D. And because of the color of the skin, they are very particular to hide their face or hide their arms and all. So multiple time we told them that okay, these things are very important. So this study is now become a very pioneer study for dealing with because our nation is a sunshine country, but still our population is 99%. This vitamin D is dependent on one of the genes which is called as VDR gene, which is inside the every cell tissue. And once the vitamin D is bind to this VDR gene, then it is now become activated to do more than 100 gene control is through this particular VDR gene. So this VDR gene, we found it as a highly polymorphic gene in our community. Still, and the other thing that we are not getting proper vitamin D. These females are the one which are having the most compromise of all those factors. Second project I would like to discuss here is like whenever I used to read when I came back to India, there is an outbreak of dengue in Delhi. There is an outbreak of dengue in South India, then Central India. I used to feel wonder that how come every 1 or 2 years there is an outbreak of dengue, and there were many efforts being taken care. So I frame a research question and design one research study, and I started working on it. So what we did, we have a mosquito population, we have a patient population and the healthy controls, and we divided Bhopal into many hot spots where we are getting those things. And we were surprised to see that everything which goes on, that which outbreak is going to happen next after a buffer time of 1 or 2 years, it everything happen inside the mosquito. And mosquitoes and these viruses are so smart, they make this particular tighter which was happened in the previous year, they just competitively displace that particular serotype and that replace with the very high titer of dengue virus. So dengue virus is basically 4 serotypes with many genotypes like subtypes. So what they do inside the mosquito, they make their titer or they make their concentration so high of that particular virus, which there is no antibody from the previous outbreak. So their outbreak becomes so pathogenic next year, next after. So for every 2 years, there is an outbreak. So what we did in this particular study, we found it very, very interesting information about the mosquito, how this evolution of dengue takes place. And we also made some prediction model that how what will happen next year in the next outbreak, which serotype outbreak will take place. And accordingly, if you do the surveillance of this type, we can also predict and tell the healthcare system that we are expecting an outbreak on particular year, you start working from January, because June, July is the time when the monsoon will be there, and outbreaks and everything, viral things will be hot and ready to spread. So this project was published in a very, very high impact factor, and I really love this work where we have done so much of studies on mosquito and dengue patients, and also the healthy control. Second, this study is not a very big study, but it's very interesting study from my point of view, because I like this study. We always see that there are people who are like, they get insomnia, they always tap their legs, and they are always very restless sort of a people. So when I started reading about this, I found there is a disease called restless leg syndrome, RLS. And I was got fascinated because it was all related to biochemistry and genetics. I thought that, why can't we have a something which can address this restless leg syndrome? So then I found it's a neurological problem, and what I was thinking was very correct. There are so many factors of chemical locha we कहते हैं ना हम लोग, so that for that, so that is responsible for this restless leg syndrome. So basically what happened in brain cell, we require dopamine, and when dopamine is required, is synthesized from tyrosine, one of the amino acids, in the presence of tyrosine hydroxylase enzyme, and they require a iron as a cofactor for this uh, particular reaction. And if the person is having deficient of iron, then what happened, that dopamine is not fully formed or partially synthesized, or sometimes there is absolutely no synthesis, and that receptor can damage the motor development in these kids, and they later on develop the restless leg syndrome and insomnia, later on hypertension, cardiovascular diseases, diabetes, all those metabolic disease and everything is like now channel. So there is some simple test, and we also try to see that if there any genetic relation. We found out that there is a heterozygous mutations which are responsible. That means one of the parent can be a carrier, and it can pass on to the infant, their next generation. So this study plays a very important role from my perspective, that we could identify the restless leg syndrome, and it is very prominent in India. We see that there are grandparents and everyone, they used to say that come and पैर दबा दो हमारे टाइप से चीजें is very common. So you know that particular thing is related to the, sometimes there is a primary restless syndrome, there is a secondary restless syndrome. But this is how this was a very interesting study which I could realize. Oh yeah, this study, cervical cancer, being a female, it's very close to my heart, this study. And when I found that whenever the female come with this disease, it was always a last stage or the fourth stage, they come to know that they are having cervical cancer. And especially in the rural areas where they are dependent on the district and the division hospital where the pap smear and other things are available, those females come to know only in the last stages. So I thought that, can I make something which we can keep in the primary health center, and they can get information about, they can test there without any cost. And these females, when I do the community round and round, if their priority is their husband is coming for lunch, their children are going to school, so they don't have their priority for their own health. So I have made a device which is like a size of one pencil box, and it can be kept at the primary health center. Doesn't require any pathologist or any main infrastructure, and any technician and ASHA worker can screen the patient, and if they get any signals for any cytological changes in the female, they can advise them. So it is a mass screening device which is now patented, and it is an innovative mass screening device which I have developed with my US collaborator. and we got more than 70% and more than 80%, more than 70% sensitivity, specificity, and more than 80% specificity for this. We are screening, and this project is still running. So this was the my outreach activity where we go to the different villages. This is my team where my colleagues are there from pharmacology, from community and health medicine, from biochemistry, from obstetric and gynae and pathology. Pathology and gynae people have helped me a lot. So we have screened almost 800 females and visited 36 villages for doing the mass screening and awareness program. So this is massive and very good. And I was like talking about the nanotechnology. So this particular work is for brain tumors, where we thought that when the brain tumors and gliomas and brain cancer are basically, their therapeutic intervention is only is radiation and surgical remove, that both of the things are very potentially hazardous for a person. They compromise the quality of life of that person. So I thought that I should recognize some markers on the brain and some medicine, so that they can cross the blood brain barrier and go directly to the brain tumors, sparing the healthy cells on the brain. So this is my next venture where I want to do the targeted drug delivery, because even the cancer patient, the chemotherapy and other therapies are extremely painful thing. So I wanted to do something that the medicine should only go to the cancer and kill it, and the rest of the healthy cells should not get affected. So for that purpose, we have developed the pilot study. Now my students are working on this. SARS-CoV-2, as I told you, the connection with this virus was from my US lab, and when the outbreak took place, me and my students worked very hard, working in BSL labs for an hours and hours, and it was very challenging working in that very extreme environment of containment. We did work on the identification, setting up the real-time PCR, and also taken up many projects during that time. They were all published in a very high impact factor. And my one of my students now, she is working as a PhD student. She is working on the SARS-CoV-2 and projecting some very good epidemiological links and how the variants are coming up, how we can address those variants, how we can further design the vaccine properly, so that we should not design a vaccine on the spike protein rather than the conserved area where they can have a n number of variants. They still, the vaccine should be potentially effective. So during this my journey of 20 years, 10 years, I haven't done anything in the science, or I was away from science. But later on, I got an opportunity and I grabbed that opportunity, and my enthusiasm to work. I got a few awards for my work, and actually the students and my MBBS, MD, PhD students are basically my strength. And I only tell them ideas. They are the one who work and give me results, and I just present them like this conference and national and international conferences. And what lessons I learned from my this journey is that whenever you start decide something, entry is always real. Barriers are there. There are people who will say that what are you doing, why are you doing this? Because when I was working in SDPGI initial days, I was not getting any salary or any designation. But you have to take something if you have passion enough for to that, and first step is always very important. There are people who used to say that if you're not a doctor, if you're not a engineer, what are you doing? Because 30 years ago, it was a very difficult for any person, because pandemic has taught us so many things, and they people now recognizing scientists also. So that time they used to say that she's not a doctor, then what she's doing in the SGPJ, and why can't she leave her job and come back and do all the activities what we do is a fun thing of. So yeah, but consistency and what I learned a lot in doing my research tenure in US, that and of course in my also, the age is not a bound. And sometimes you have to keep your ego, because when I started, the students were junior to me, and how to be in their team as a student at that time I started after 10 years, what was really tough. But I adjusted, they also accepted me, I also liked them very much. And mentors are definitely, you have to find a good mentor, and mentors are there, but you have to identify, and you have to trust them and work in that way, that that they ultimately going to support you. And the learning thing for me is that we must do what we actually love to do, because when you get this opportunity, what you are actually passionate about, and when you do, you just be in your own world. So, with that, I would like to thank everyone. And that was just a small, brief information about my journey. And I thank all my railway family for being so supportive during these years to me and Nitin at every front. And thank you. Life was probably with my presentation doesn't look like that tough, but it was definitely so many downs and very so little up, and very challenging. But at the end, I say that all God's blessing, I am happy. I'm doing what I really enjoy doing. Thank you very much.
Thank you very much, Dr. Rashmi Chowdhary, for sharing your journey. It is a very impressive and your
Thank
credentials
you,
are
sir.
very impressive. I
Thank you,
think
sir.
the range itself, from polio to H1N1 to dengue to molecular biology for cancer research and nanotechnology. I
Yes,
think
sir.
you're really at the cutting edge of research in this field.
Yes.
And thank you for sharing this journey with us. It has been very enlightening,
Thank you, sir.
and really it shows us that you can begin at any age.
Yes, sir.
That's a very inspiring story by itself. So thank you once again, and thank you everyone who has joined in.
Thank you, sir.
I would,
Thank you. Thank you.
if you agree, we can ask if we
Good
can ask
evening.
participants if they
Yes,
have
Good
any
please, sir.
questions.
Good evening. I'm R.K. Jain, 56 batch.
Namaskar,
Rashmi,
sir. Namaskar.
Rashmi, it has been tremendous pleasure to listen to you. The great strides that you made and wonderful things you do. Although much of what you have done research and goes much above our head because it's not our area of understanding, but you have done a great job.
Thank you,
It's been
sir.
a
Thank
pleasure.
you. Thank you, sir. Thank you for your encouragement.
I'm reminded of my wife's own this thing. 20 years after our wedding, when I was posted at DLW, she decided to
Oh,
continue her study, and she did research and finally got PhD from BHU in
wow.
in economics and genealogy because she was
Oh my
interested in
God. What a tough
various.
subject. Yes.
So that reminds me what a person can do if he or she decides to do, irrespective of the age. And that's one
Yes.
thing.
Yes.
And one thing which I noticed that you have done some research in restless leg syndrome. That's one thing which my wife keeps on complaining about it. And
Oh,
वो कुछ ना कुछ उसको दवाइयाँ दे देते हैं कि गैबापेंटिन ले लो या
yes.
कुछ इस तरह के ले लो. But
Yes, sir.
anyway, That's something which you have done some research, and I'd like to get in touch with you later.
Sure, sir. Definitely, sir.
But overall, it has been a pleasure, and I'm extremely happy that you have done such a great thing. Thank you.
Thank you, sir. Thank you, sir. Thank you. It means a lot to me. Thank you very much.
Yeah, Dr. Rashmi, thanks very much. It was a great talk. And I love the lessons you brought out at the end. Especially learning is not age-bound, is ego-bound. Very well brought. This and many other lessons. Well done, really. Starting out, without any salary or designation, I think that was a big breaking out for you. Terrific. Thank you again. All the very best.
Thank you. Thank you very much, sir.
Rashmi, this is Anil Khurana, 1978 batch. I live in Boston.
Okay,
and
sir.
really delightful to hear your conversation, both in terms of your passion, but also the extensive depth and breadth of what you've covered. Very impressive. I know a little bit about life sciences, and I can certainly say the breadth and depth are beyond what I would have expected of any normal human being. So a lot of effort and passion from you. So kudos to that. Maybe a question, and this is a naive question in some ways. You covered many treatments and studies. How are you commercializing this? And when I say commercializing this, clearly there's a financial aspect of that, but I'm not talking of that necessarily. I am though, but also making it available to larger number of people.
Yeah.
Right? Because if you publish a study and 10 people read it, and 25 people like us naively listen to it, the impact is not yet seen. So what's the commercial approach that you're taking to bring these to market, to customers, and so on?
That's a very important question. Actually, this question was asked by the ICMR Director General also very recently when he visited. He said, whatever you are doing on the academic front is okay, but it should reach to the patient. So for my clinical, for cervical cancer device, we have patented it, and now we are into the clinical validations. Once it is done, we are going to have an expression of interest, we will call it, and we will do commercialization. Those things are taken care by our AIMS PRs, the IPR cell. They do the commercialization, and we have to do with their agreement only. Yes. So definitely you will see those things with the ICMR, and so we created this device, and now it is open for clinical validation, and it will go for commercialization very soon. Yes.
Okay. No, that's great. And I hope that institutes like the AIMS, Bhopal, and other ICMR have the skills and capabilities to do that, because that's a key important capability as well.
Yes.
And just as a side note, and we can take this offline, one of my friends who is in Hyderabad, he's actually based in Hyderabad and New Jersey. He's a software engineer, but he has a cervical cancer treatment approach that he's working on as well. So if you want, I can put you in touch with him and you can take it from there.
Yeah, sure. Definitely, sir. That will be very good. And we will try, I'll share my email ID or my phone number. I think Nitin must be having your number. We will be in touch, and we will definitely go in, because this is a need and of an hour of this cervical cancer. It is 100% treatable cancer if females are diagnosed in early stages. So I think it's our duty and a moral duty of every scientist to work on such type of disease which are curable. We should pay attention towards it. Yes.
Yeah, thank you. Once again, congratulations. Wonderful to hear that.
Sir. Thank you very much, sir. Thank you.
In fact, you remind me of my own journey from a railwayman to a IIM professor.
Wow.
And that too teaching literature of all the things. I never dreamt of doing that.
Yes,
so anyway, it was good. And one point you made is that if you love what you are doing, then that is actually a great blessing, because not everybody can get that kind of an opportunity.
Yes.
So now are you working on Ebola virus by any chance?
No, sir, because Ebola is not in India. It's touch wood. It's not there right now.
Yeah.
So I'm not. But yes, we are like health ministry must be taking care of it. And all those studies which we are doing for epidemiological studies or identification, those things ultimately make a very much difference. And I hope this particular virus should not come to
Right.
India by any chance. Yeah.
Yeah, that's true. I hope doesn't actually.
Yes, yes.
But did you have any chance to meet Dr. Gagandeep Kang by any chance? You know, she's famous, of course, you must be knowing her.
Can you sir repeat the name again?
Gagandeep Kang, you know, she is also
Oh
the
yeah.
daughter of
Oh my
GSK
God.
Kang 49
She
batch.
is, oh my
Yeah,
God.
fellow
She's
of the Royal
our role
Society.
model. Yes, yes, yes. I met her during conferences and when I was studying in and working with WHO at that time. So in many WHO meeting and all those things, I often get an opportunity to see her from distance because I was so junior at that time. I was just studying. But yes, I really admire her a lot, and she's like just like my role model in the virology. Yes, I know her very well. I appreciate her work a lot. Yes, sir.
Rashmi, just a personal question.
Yes, sir.
What had you studied before you landed up in Lucknow at SGPGI? What
Uh,
had you all been through already?
Yes, sir. Sir, I was into the life sciences and I was doing my master's that time. And
Okay.
when I came to Lucknow, it was already 10 years gap of my post graduation and my this thing. So when I started, I just started from the scratch actually. So life sciences was my stream. Yes, sir.
Well, you had a master's in, say, life
Life
sciences?
sciences, and also I did my master in philosophy, that is MPhil degree. And after that, in the molecular biology, that was my core area of specialization. And after that, I got an opportunity later on, after 10 years, to do my PhD.
Great.
Yes, sir.
I'm
Thank you.
I'm certain your background in molecular biology at master's level would have
Yes, sir.
helped you in getting into this. Otherwise, I don't think it's easy for anyone to get into the laboratory and not to be starting from scratch is very difficult. I
Yes, sir,
think
it
you.
was. Actually, it was. That's what I'm saying. I'm just talking all the good things in my presentation. But actually, it was not that easy. It was very difficult. If someone tell me now that can you relive all those things again? I said, no, I don't know who was that girl who was doing. I am totally different person now. I don't know how come I have done all those things. It was difficult now to rethink and go and relive. I cannot do that. That was a different state of mind. Yeah.
Dr. Rashmi, I think you have done a fantastic job. It is really a pleasure to hear you. And in fact, I was waiting quite eagerly for this because
Oh, thank you,
from
sir.
Nitin, I had known some of your accomplishments.
Yes.
Um, very great. One question that I had was that, you mentioned the research scene scenario in India. You started with the PGI Institute, then you moved to the US. And you said that the research scenario in the US is very different from India, and I expect that it is much more mature. But now, do you see some improvement in the research scene in India regarding immunology, virology, etc.? Are there more institutes opening up? Are there more BSL labs opening up?
Yeah, definitely. I mean, there is a so much change after the time I left India. We were like very hard crunch on the budgets and all. Even setting up a small experiment, we didn't have any machine or the reagents which were required to set up that experiment. As compared to that, when I moved to US, they just wanted ideas and the fund was never a problem. They said you set 10 experiment. If one works, that is good enough. And the thing that what I learned from US that they never hesitate in testing. In India, if you write some project, there are 10 people who will say that this won't work. This is something not a good idea. So now what is that after COVID? funds is become, the labs have been equipped, the infrastructure has enhanced a lot. So there are people working, and we are also having a private participation is increasing. So funds has become more easy, and ICMR and government has given an opportunity to write up more, and more calls are there to share the ideas. So there are early stage call, there are small research grant, there are intermediate research grant, there are huge research grants for like 50 to 100 crores rupees of funds are provided. So infrastructure wise and opportunity wise, definitely we have come a long way, and now we are more focused, because for aims this is a very important uh, pillar of aims. Or none of the state medical college or any other we will have this research apart from hospital. So our doctors, young doctors, budding doctors, everyone is into the research. So that is the beauty of being in AIMS Bhopal. Yes.
A related question. I mean, you mentioned funds are being made available by the government. How about the private sector, the pharma companies, or the privately funded institutions? Are
Yeah,
they spending money on such projects?
that
Uh,
is I mean comparative good enough. And they also can, I mean, there is two types of research. That first, we can have of government funding. We can also apply for some private funding like Wellcome Trust grant, Bill Gates Foundation grant, and all those stuff, BIRAC and all. So those things, those are also funding a lot. And the focus is now everything on the translational research. Earlier the research was used to be like that, I have found four component, and one are increasing the immunity, one is decreasing the immunity. That research was only into the documents and the publication. Now government wants it to be on as a translational research, so it should reach patient. So for that purpose there are many ideas which are floating. People, even the first year student, they are making devices for early detection. They are working on AI, and they are doing lot of things are going on. It's totally different scenario. I cannot even imagine I could have think of such things when I was at 10 years back. Now the kids are like onto doing lot of thing using AI and other thing, and they have lot of opportunities also. So
Great, thank you.
definitely we are growing
Can I,
and we are doing
can I,
good.
can I ask one question?
Yes,
Was
sir,
COVID
please.
was, was COVID was COVID an engineered virus, or was it a natural?
No, sir, it was natural. It was natural virus. It was not engineered virus. Coronavirus has this tendency to evolve. They have this tendency to, they, whenever there is a pressure, they evolve. All viruses are like that only. They evolve against the pressure. As I told you, dengue virus, they are so smart. I mean, how can they manipulate themselves in mosquito, and they find their host, their vector so intelligently. So coronavirus have this history of evolution. First SARS-CoV virus, one was in China, then in Arabic country, Saudi Arabia, then this was the third one. Yes.
Thank you.
Namaskar, sir. Keshav Chandra, sir. Thank you, sir.
Yeah, wonderful, great. I really respect your passion for this work and how you have really come up so ingeniously on so many fronts. Amazing.
Thank
Like
you, sir.
Rashmi and all our friends and seniors here, in my own very small way, on the sidelines, I've been into voluntary blood donation, and that I've done since 1976, and I've done it 85 times. But that is incidental, Rashmi. What is more important is that my policy promoting healthy voluntary blood donation was accepted by Government of India in
Wow.
2019 October. So, of course, you've made a great talk today, and of course, with Nitin by your side, who has worked with me both in RDSO and
Yes,
board,
sir, you
I
saw
can,
us
I
last
can
struggling
vouch,
days.
that the two of you make a great, brilliant pair.
Yes,
Beautiful.
sir.
God bless
Thank you,
you.
sir.
I
That's
really
God's blessing, sir.
great.
God's blessings.
Thank you, sir. Thank
Wonderful.
you for your kind words, sir.
Right, Nitin. God bless you. Great. Nice
Thank
to hear you.
Yeah, sir.
Hello,
You
sir.
know our struggling days. You were there in Lucknow that time. Yes, sir.